this chapter, we address the control and destruction of microorganisms, a topic of immense practical importance. Although most microorganisms are beneficial, some microbial activities have undesirable consequences, such as food spoilage and disease. Therefore, it is essential to be able to kill a wide variety of microorganisms or inhibit their growth to minimize their destructive effects. Microorganism control continues to be a hot topic as microorganisms evolve to resist current strategies. Control efforts have a substantial role in public health to prevent disease as well as in therapeutic use to treat disease. The growth of microorganisms can be controlled by physical, chemical, mechanical, or biological means. In general, any chemical, physical, or biological products that controls microorganisms is referred to as an antimicrobial agent. First, we need to define the following key terms related to microbial control. Sterilization is the removal or destruction of all living microorganisms. Heating is the most common method used for killing microbes, including the most resistant form, such as endospores. A sterilizing agent is called a sterilant. Liquids or gases can be sterilized by filtration. Control directed and destroying harmful microorganisms is called disinfection. The term usually refers to the destruction of vegetative or non-endospore-forming pathogens, which is not the same thing as complete sterility. Disinfection might make use of chemicals, ultraviolet radiation, boiling water, or steam. In practice, the term is most commonly applied to the use of a chemical, a disinfectant, to treat an inert surface or substance. When this treatment is directed at living tissue, it is called antisepsis, and the chemical is then called an antiseptic. There are modifications of disinfections and antisepsis. For example, when someone is about to receive an injection, the skin is swabbed with alcohol, the process of degerming or degermination, which mostly results in the mechanical removal rather than the killing of most of the microbes in a limited area. Here is a table listing all the related microbial control terminology. Decontamination is to distract, remove, or reduce the numbers of undesirable microbes. Asepsis, disinfection, sanitization, and degermination are all methods of decontamination. Sepsis means the growth of microorganisms in the tissues, such as blood infection or infected wound. In correspondence to sepsis, asepsis are techniques that prevent the entry of microorganisms into sterile tissues. Aseptic techniques are important in surgery to minimize contamination from the instruments, operating personnel, and the patient. The resulting terms are quite familiar to you as I have just mentioned in the last slide. When bacterial populations are heated or treated with antimicrobial chemicals, they usually die at a constant rate. For example, suppose a population of 1 million microbes have been treated for 1 minute and 90% of the population has died. We are now left with 100,000 microbes. If the population is treated for another minute, 90% of those microbes die, and we are left with 10,000 survivors. In other words, for each minute the treatment is applied, 90% of the remaining population is killed. If the death curve is plotted logarithmically, the death rate is constant, as shown by the straight line. Several factors influence the effectiveness of antimicrobial treatments, including the number of microbes, nature of microbes in the population, temperature and pH of environment, concentration or dosage of agent, mode of action of the agent, presence of solvents, organic matter, or inhibitors. Here are some examples of the factors that influence the rate at which microbes are killed by antimicrobial agents. Figure A illustrated the length of exposure to the agent. During exposure to a chemical or physical agent, all cells of a microbial population, even a pure culture, do not die simultaneously. Over time, the number of viable organisms remaining in the population decreases logarithmically, giving a straight line relationship on a graph. The point at which the number of survivors is infinitesimally small is considered sterilization. Figure B reveals the effect of microbial load. Logarithmic plotting, the blue lines, revealed that if the rate of killing is the same, it will take longer to kill all members of a larger population than a smaller one, whether using heat or chemical treatments. Figure C showed the relative resistance of spores versus vegetative forms, while Figure D showed the action of the agent, whether microbicidal or microbistatic. As we can tell from Figure C that spores are more resistant to antimicrobial agents than vegetative cells. Here we summarize that prions and bacterial endospores have relatively the highest resistance to antimicrobial agents, while Pseudomonas species and Staphylococcus aureus have moderate resistance. Most bacterial vegetative cells, fungi spores, yeast, enveloped viruses, and so on are more sensitive to antimicrobial agents, exhibiting the least resistance to antimicrobial resistance. This table gives some example of the relative resistance of microbial endospores and vegetative cells to control agents. For instance, endospores are more resistant to the pressure than the vegetative cells in any kind of forms, including heat, radiation, sterilizing gas, and sporicidal liquid such as 2% glutaraldehyde. Here we summarize the microbial control methods, and these focus on the control of microorganisms by physical agents, chemical agents, and mechanical removal methods. Physical agents covers heat, whether dry or moist, and radiation such as ionizing X-ray and non-ionizing UV light. Chemical agents include both gas and liquid forms. Mechanical removal methods indicates the filtration of either air or liquids.